So good afternoon, everyone, and a very warm welcome to this inaugural lecture by Professor Richard Mellanby in honour of his recent appointment to the Chair of Comparative Medicine at the University of Edinburgh. And he's proving his qualifications for comparative medicine, as you can see on his title, referring to dogs, cats, mice, sheep, penguins, and one monkey. So we'll look forward to hearing more in due course. So my name's Moira White. I'm head of the College of Medicine and Veterinary Medicine, and I'm here on behalf of the principal, Peter Matheson, who is away on holiday. I've realised I've left you all standing. So maybe you'd like to sit down, because I am going to carry on for another minute. So, my pleasant duty is, as well as welcoming Richard himself to give his lecture, to welcome members of the family, his mother and father, Anne and David, his wife Katie, his children, Hamish, Poppy and Sophie, who are looking interested at the moment, so keep it up, and his mother and sister-in-law, Annabelle and Celia Broadhurst. So thank you all very much for coming. I don't know who's responsible for Richard's very smart turnout today.
It's always like this.
Always like this. So, Richard, as many of you know, is a graduate of Glasgow Medical School, but left there shortly after qualifying and migrated south to Cambridge, where he was a Wellcome Clinical Fellow, before coming north to Edinburgh, where he has now been for about a decade. Richard is, to my mind, a model clinical academic. He is highly regarded for his clinical practice. He takes teaching and training very seriously and has been an integral part of the ECAT programme, the Edinburgh Clinical Academic Track programme, which trains the highest calibre future clinical researchers, both medics and vets. And he also has a very successful research career, as evidenced by his Wellcome Intermediate Clinical Fellowship, which was extended by the Wellcome Trust, and has pursued research in a number of important areas. And initially I thought maybe he was going to talk about his valuable work in sub-Saharan Africa, reducing rabies, but I believe he's going to talk about his work at the interface between metabolism and the immune response, which is a very important and growing area of research. So, Richard, we're looking forward to hearing what you've got to say.
Thank you very much. Thank you very much. I'm very mindful that Professor Argyle told me he's got a steak tonight, so I've got to make sure this is on time and swift. So if any of you are feeling restless, just let me know and I'll get a move on. So as Moira said, it's my sort of great honour and privilege to work across the campus, both in the Hospital for Small Animals and more recently since my move from the Centre of Inflammation through into the Roslin. And it's just a tremendous honour and privilege to have this opportunity to work between these two really fantastic facilities within the university. As Moira said, my research interest is very much focused in around nutrition, inflammation, and health outcomes. And I try to take with our research this very integrated approach where we try and understand diseases that we see in the patients that come in through our hospital, you know, and we'll talk a little bit about some of the patients that have really influenced our thinking over the years. We then try and study the disease processes or the biological processes that we're interested in big population studies, and hopefully this is 150 towards your 250,000, Bruce, today, if we get a few mentions of big data for you. And we then search this mechanistically from a process point of view with tractable experimental model systems. And what almost everyone does that has the opportunity to do this sort of gig has to try and persuade their audience that what they do is of some interest and of some wider relevance, and ideally summarise that in one slide. And this is the one that I tend to wheel out for these sorts of events. This is a screenshot from the BBC website about a year ago. Some of you may be aware that we had a referendum recently. It was the Brexit discussions, and so this was when May and Merkel were trying to work out what was going to happen to the universe. In this time, we also had the nucleus collision. We also had Labour with this sort of sense of disarray. But what topped the news on the BBC website is just our national obsession about vitamin D and about what its importance is towards our health, and particularly in the non-skeletal health aspects, how much should we be taking, and does this genuinely influence long-term health outcomes? And this is still a huge public health debate that my research tries to inform. Now, hence the title. The second Mellanby I'm going to introduce in this story is Sir Edward Mellanby. Now, the Mellanbys are a very small clan. There's about 120 of us, and almost all of us come from Hartlepool in some description. So both myself and Sir Edward were born there. And Hartlepool, as many people know it as the Riviera of the North, mainly by people who have never been to France. This is my go-to slide for that, and I think it's well, one of the nicer shots that you find on the web because there's no graffiti and all windows are intact. But I suspect that's more the power of Adobe Photoshop than reality. But even in this, you can see our historical legacy hiding away in the background there. Now, we don't have many famous people come from Hartlepool. I'm not sure if anyone recognises this fella. I was worried when John Savile wasn't here. He was going to be our go-to man for this little segment. I hope he blurted out the answer without being able to control himself. But this is Phil Middlemiss, and he broke a lot of hearts in Coronation Street. So whenever we have a sort of Aldi or Lidl that needs opening, or drug rehabilitation centre or something like that in Hartlepool, he's absolutely our go-to man. The only other person of any real note that had a connection with Hartlepool was Ridley Scott. So Ridley Scott was famous for, well, he spent a few years at Hartlepool Art College and then went on to have this incredible career as this sort of iconic movie director. And his famous work was Blade Runner, which I don't know if many people have seen this, but really it's this unbelievable post-apocalypse vision of what hell might actually look like. And the images of despair and deprivation was something that really caught the sort of media interest in trying to understand, well, what influenced this? And the astute amongst you might know where this is heading, and this is what he went on the record as saying. Scott has revealed how his nightmare vision of the future was based on Hartlepool's filthy industrial landscape. But the key thing here, and the key thing to take away from this, is that we in Hartlepool are very upbeat. It's changed beyond all recognition, and for people that live in Hartlepool, this is hard to believe. This is very much the party line. It's not really reflected in reality. But I hope with the big screen, because like all scientists, I only use the most reputable of information sources, Wikipedia, for an inaugural, which, yes, now perhaps it doesn't seem like such a great idea. But this is the monkey of the story, and what Hartlepool is most famous for is our relationship with a famous monkey many years ago in the Napoleonic era. So the fisherman from Hartlepool caught this monkey, and because it wasn't something that he had seen before, assumed it to be someone from France, and then went and hung it. Was the top and bottom of the story. I think one of the things that gets lost in this sort of quaint little story is that we gave the monkey a trial. So, you know, we're not the land of savages in Hartlepool. The monkey has an opportunity to have his say. And legal scholars do debate how easy it was for the monkey to put up a defence. What you're doing on the boat, young'un, and all of this sort of Hartlepool vernacular. But the monkey got a trial. Now, many sort of communities would just say, oh, well, it's a funny little story, and it was before the internet, and we didn't really know any better. But actually, what we do in Hartlepool, we really truly own this story, and that's the glorious thing about Hartlepool. Because this just comes down in Wikipedia a little bit later. So, monkey hangers or chimp chokers, the abuse we receive as Hartlepool United supporters before there's acts of mass violence on the pitch from Darlington rivals. But the mascot of Hartlepool is Hangus the monkey, very amusingly named. And the man in the monkey costume, Sir Stuart Drummond, stood for the post of mayor as Hangus the monkey and campaigned on a platform which included free bananas for school children. To widespread surprise, he became the first directly elected mayor of Hartlepool, winning 7,500 votes with a 52 stake in the vote at a turnout of 30%. And brilliantly, he was really turned on the next election, where he won by a landslide with this mass turnout, a turnout which really sort of mirrors what we get in general elections. So what's brilliant about this is the fact that he got free bananas. It sort of implies there was a sort of progressive left-of-centre policy that he was trying to bring in. The only thing he really stood for in his manifesto was the free bananas. And that was really a bit in the balance as to whether that was truly going to happen. And what's also quite nice about this is, to widespread surprise, if you lived in Hartlepool, the idea that we would elect in a man dressed as a monkey suit is no surprise to you at all. This is entirely in keeping with the community ethos. But I think below the surface there's a different story to tell, and that story is often wheeled out to the dangers of democracy and, you know, why giving the working man a vote is always a bad idea. But actually I think the sort of true story is slightly more subtle and nuanced than that. And this is just some unemployment data figures from the region, where you can see in the northeast we struggle with unemployment, and particularly in Hartlepool we struggle massively with unemployment. And so when the New Labour project started in '97, and we had this sort of huge air of optimism within the North East that the challenges of the Thatcher era were beyond us, and we were going to have this sort of slightly more community, progressive left party brought in. And basically, the top and bottom of it is it didn't really quite transpire. And our local MP is Peter Mandelson, or was at the time, and he was really the architect of all of this. And many people would argue he's an entirely reasonable MP, but many people would also feel he was massively disconnected with the local community that he served. And that was most infamously caught when he went into the chipper in Hartlepool and asked for some guacamole with his fish and chips. And it was mushy peas, bonny lad, was the reply. So they dreamed up this plan, him and Prescott, to basically have devolved democracy within the North East. And so we were going to have this mayoral system. And he was very, very keen to to get on board one of his Labour Party stooges. And so really this wonderful grassroots movement emerged upon which everyone in Hartlepool was galvanised to vote in the monkey against Mandelson's stooge from the Labour Party. And so really this wonderful sort of campaign developed because they just wanted to create this moment, this one photo, this one image by which the most pompous man in Westminster politics was made to stand next to a man dressed in a monkey suit whenever he did his civic business. And this is what we created. And so for all of those that are slightly frustrated with our political representation, we have much to teach you in Hartlepool. If you actually did what we did in Hartlepool, we perhaps wouldn't have this fella ruling the free world, and we perhaps wouldn't be standing over the abyss that is our current Brexit vote at the moment. And ultimately, the people of Hartlepool brought down this scheme because Prescott was so worried that the tiger, that is the Hull City mascot, was going to be voted in as Hull's representative. He just kiboshed the whole scheme. So anyway, back to Sir Edward. So back in Sir Edward's day, rickets was a huge, huge problem. In Edinburgh, the children suffered with sort of choking on silver spoons, but in other parts of the country, particularly in Glasgow, rickets was a huge problem, with 50 to 60% of all kids having this really marked disability that created huge long-term sequelae for the affected children. And so the entire history of the vitamin D story can be traced back to this rather innocuous letter from Sir Walter Fletcher, who was the first head of the MRC. And he wrote, basically passing on a message that he had with Hopkins, who's Frederick Hopkins, who was awarded a Nobel Prize for his work on vitamins, twisting the arm of Sir Edward to move away from the work that he was currently doing on creatinine metabolism to start to think about doing some work on experimental rickets. And it was suggested there that maybe he wanted to work around the £200–250 to begin this work. And it was a bit of omen and error, and I don't think Sir Edward was very keen on getting on with this, so it took him till September to craft this extensive grant documentation that he put forward here. And in classic Mellanby style, although he was told to top it at maximum 250, he went in at 300. And this is a trait you learn in the crib as a Mellanby, that you can always negotiate down but never up. So this is very much in Sir Edward's mind when he started this. He gave a very, very detailed account as to how he was going to spend the money. And then he's already laying the foundations here about saying, this could take a bit of time. And it could also be a disaster. But if you want me to do it, I'll crack on. So there was this wonderful exchange, and it came backwards and forwards. Here he also talks about some alcohol work, which we'll come on to towards the end. And he basically says that things are still progressing. And then ultimately, this real landslide piece of work was published in 1919, which I would argue is one of the most important pieces of public health research done in the interwar years. What was really lovely about this is it really, on this one figure, really captures the beauty of experimental design. And what we have here, we have dogs being fed bread and milk and linseed oil, and on this one, we have them fed exactly the same bread and milk, but with cod liver oil instead of the linseed oil. And what you see here in this sort of beautiful, simplistic approach was that the dogs that got the cod liver oil, they were protected from rickets. And from this point on, the world really changed in terms of the public health for this skeletal abnormality. Images like this became commonplace up and down the country, with children being fed cod liver oil. And by the 1930s, it was impossible to do clinical research on rickets because the disease had really disappeared. And many companies, including the Wellcome Trust Drug Company, made a small fortune out of this, which is something I'm very keen to remind them of every time I apply a grant for them. So Edward's career went stratospheric. 6 years later, he was awarded the Fellowship of the Royal Society. He got a whole load of other accolades, including the Harveyan Orator a few years later on. And I think this is the one in his archive that really caught my imagination, because I think if you're on an Argentinian nutrition journal, just reading a book and looking very clever, you've cracked it. You know, your work on earth is pretty much done, and that's about as good as it can come. So he was a man who was a tremendous, towering figure within biomedical research in the early part of the 1900s. So for those of you that are still awake, you will understand there's something of a slightly different trajectory between Sir Edward and myself. And this has been made clear to me on many, many different types of occasions. Perhaps most memorably last year, where I spent an inordinate amount of time working out how to wash dog bowls within the hospital. The dog bowl washing machine had broken down, and Shante had asked for £3,000. Getting £3,000 out of Tim King for a dog bowl washer would be like trying to cure the Middle East. It would be a very difficult negotiation. So I became encyclopedic in the justification for how we had to wash, what temperature, this sort of thing. And at the same age point, really a few years older, to be fair to Sir Edward, he was basically running the national research agenda as head of the MRC. So there's huge similarities, our surname, our place of birth, our research interests, our formative time at Cambridge. But like every good student, when you start to think, well, where did it go wrong? I think where it went wrong for me is I spent, rather than going straight from Hartlepool to Cambridge, I took a detour of 9 years through Glasgow and Newcastle. And with these very fine, magical cities, there's an awful lot to distract a young man. And when I think as to the places, and I think very, very fondly of my time in Glasgow, these would be the places that would be the main beneficiaries of my student grant. The Hive tenants, which I basically bankrolled with my friends, I would have thought, over a long period of time. And when I put this together, I was struggling to find a picture of Clarty Pats because it's changed, but I was one of an elite group of athletes that managed to go to Clarty Pats 4 nights on the trot. And I reckon there's more people have swum the channel that have achieved that. It's, the chances of doing that and not being chucked down the back stairs is 1 in a million, I would argue. Now, I had a wonderful time at vet school. I was taught by some of the greats, including our very own Professor Argyll. But I was also taught by this fella here, who I'm not sure if anyone recognises, the evergreen Tim King. So Tim King is a wonderful man who does a huge amount of good for the campus behind the scenes and just making the place work. And one of his roles with this practice, where he ran what can be best described as an eclectic group of individuals, was really just to bring into the clinical practice the things that we were being taught at university by people like Professor Argyll. And he did this job extremely well. In our yearbook, you have to try and capture, well, what did you learn from these individuals? And this is what we learned from Tim King. So him and Professor Argyll used to work hand in glove on the translational oncology agenda. But because Tim King was such a legend, we did learn something else from him. And this is the other thing. And I'm pleased to report that, I don't know if Tim's here, but your teaching lives on, brother. This is something that we're, again, we're very aware of this mantra that you taught us all those years ago. But my real formative time really happened in my intercalated years, where I was the very fortunate recipient of a Wellcome Trust Intercalated Award, which allowed me to spend 2 years in the Graham Kerr building. And this was just an incredibly wonderful time, because you don't really have to do an awful lot of work as zoologists compared to being a vet student. And we had these absolutely massive summers off, upon which I went and did a whole load of conservation work and got my first taste of research. And of all the sort of junk that we variously throw out, and Katie tells me more needs to go all the time, this is one file of letters that I have retained, because it's just a wonderful memory of some of the work that we did back there. And, you know, as this sort of boy from Hartlepool, I wrote all these letters saying I was prepared to take a group of people to do some biological conservation work. And all these letters from all over the world flooded back with ideas of what we can do. And this was one from Roland Worth, a guy who I've never met from the IUCN, who wrote this incredibly extensive letter of all the things that we could do. We could go and try and find these rare primates in Vietnamese markets, these fruit bats in the Philippines. And suddenly the world just suddenly seemed to open up. And the letters came in from absolutely everywhere. We had letters in from Ghana, we had ones from Mongolia, we had ones from Pakistan, Turkmenistan. They came from absolutely everywhere. And again, one that stuck in my mind was one from Yemen that encourages us to go, which is rather nice how it turns the full circle, to go and study rabid dogs within Yemen. Now, I'm sort of very keen to study the disease with the highest case fatality of any zoonotic disease in a baking hot country with a very equivocal human rights record. I was keen to do that as the next man. But when the invitation to the Seychelles landed, we had a quick appraisals meeting in tenants, obviously, and we decided we'd go there. And I'm very, very pleased to report it was absolutely every inch the tropical paradise the bounty adverts would have us believe. It was absolutely wonderful. And to my immense amazement, given the quality of the people I put together on this trip, we actually did quite a lot of vaguely useful work. We studied the fruit bat, the Seychelles fruit bat, this magnificent creature here. We mapped where they lived, we worked out their threats, and we did a little bit of work on their ecology. We also studied again this magnificent animal, the Seychelles white-eye, which is one of the most threatened birds in the world at the time. That was known only for a few breeding pairs, and we were able to find more and increase knowledge about this population. And we were then able to visit this island to the north of the Seychelles, where we basically leveraged on the fact that a study had already been done here about 20 years ago, where there was lots of people living on this island. They did a bird census. The island depopulated because the industry they were involved with had just basically collapsed, and we went in there and did another survey. And what we were able to do, and it was this wonderful, hopeful story, that is basically when humans move out of an area, the endemic birds can then start to flourish again and outcompete some of the invaded birds that had arrived there. So it was a wonderful story of hope and of optimism that actually, in the biological conservation story, that animals can, that habitats and ecosystems can regenerate if humans leave them alone. And this was the very first paper that I published. And after we did this, right from the start, from inception of idea through to execution of work, through securing of funding and all the rest and all the ups and downs that entailed, to see the end output, I knew that this is what I wanted to do with the rest of my life. And I hadn't really quite worked out all the details yet, but this process, which I still think, and again in all its ups and downs, is a wonderfully joyous thing to have an opportunity to be part of, is something that I started with that paper. It was also my first taste of leadership, and I hadn't got my diploma from sort of person management, HR and union negotiations from the Argyll School of Leadership at this stage. So I was somewhat wet behind the ears, and in a sort of twist that you wouldn't even really expect from the scriptwriters of the In-Betweeners, next to these 6 young men in our house in Seychelles, moved in 6 girls from an all-girl college in Oxford that moved in right next door. And it's fair to say productivity in both groups plummeted once this sort of housing arrangement started. And to my eternal shame, one of our group actually asked in one of the sort of town hall meetings that we would have whether he could have a few days off his work to consolidate his relationship, which is a wonderful euphemism, to which I shamedly said yes, because he was so love-struck, I just couldn't stand it anymore. But look at the monster that you've created. We wouldn't be having this now. And so basically the projects from this just followed on year after year once I'd got the hang of this. We went to the Seychelles, we went to Ecuador the following year, where we worked in this montane rainforest. And again, it was just a wonderful, wonderful opportunity to visit South America. We went to Pember Island the following year, where we studied the rare birds within the Nagisi forest. And probably the most memorable of all the trips, and one of certainly the absolute hardest that we did, was when we went to try and find the slender-billed curlew in Kazakhstan, which is this amazingly rare bird, which is now widely considered extinct. And we took together really a huge team of world authorities on this from all over the country, from universities of Aberdeen, Oxford, Edinburgh, people from overseas, people from NGOs, all came together along with host country scientists to try and find it, as we drove 10,000 kilometres around Kazakhstan and these dilapidated old ambulances on a sort of poor man's cannonball run. We were driven around by these Marmite-loving pirates. We had to work within the appalling infrastructure that was just in this sort of collapsed Soviet state. And we just got into this rhythm of working really many, many days of hard travelling behind even running water. Ludicrously dangerous looking back, but nonetheless at the time incredibly exhilarating, and a time upon which we just had found our niche as a collective group that we were organising this to take these ideas from start to finish and publish widely on it. And this really was the pivotal time in my life when I knew that what I wanted to do was science. But I also knew that I wanted to get into vet medicine. I wanted to, in essence, consolidate what I did and had learnt about, and this is from the glorious time of our time in Westways. Now, whenever we get together with friends from this time, my wife again reminds me how incredibly boring these stories are, and I'm on a very strict no ad-lib gig tonight. But it was a time of just extraordinariness, really, and I think that was the thing that really just was so striking about this, is just how extraordinary and how ordinary this all then just became. And that was really, I really remember this time when I went into the practice, and I saw this 30-kilo German Shepherd dog in a Silver Cross pram, designed for about an 8-kilo infant. And it's pretty weird. But what was really odd about this is that no one thought this was strange. And I went to tell my boss, Jez, and I said, there's a dog in a Silver Cross pram. And he goes, well, yeah, no, it's fine. Yeah, I saw that last week. I'll just go out and have a word now. And it was just, and that just was just the way it sort of worked. The extraordinary became the everyday. One of my very first house calls was down to the bike, and part of the issues here is we just serviced a very wide range of the community, and down into the disenfranchised and sort of slightly challenging areas of the biker. I went there to do a house visit and came back slightly ashen-faced, and Jez called me in and gave me a bit of a sort of telling off about the client who'd complained then about my sort of poor bedside manner. Who could have thought that, eh? But it turns out, when I tried to explain to Jez, well, he had a gun, Jez, and I was somewhat rather nervous about the scenario, and I thought maybe we could pick up on the erythropoietin axis and discussions of that when he came into the surgery without his gun. And so maybe that was the sort of thing that was making me slightly unnerved. But then the flip side of that, we also served Jesmond, which is an incredibly posh part of town. And so my very first significant surgery that I did out of hours was to an emergency cesarean on the dog that was owned by the head of the IVF treatment centre in Newcastle. So that was kind of harrowing enough that all of that was interplayed, but what made it even worse is that she made this request that she and her extended family could sit and watch, which was very, very harrowing. And also I was doing it with a nurse who basically had just been elevated from receptionist 2 nights before, and this was the first anaesthetic that they'd ever done. And so rather comically I was saying, well, she said, well, where's this tube go, pet? And I said, on the endotracheal tube, and that's what's keeping the dog asleep when I have my hands in its abdomen. And so all of this sort of horrendousness just played out almost on a daily basis. And so really after a few weeks of this, my adrenals were the size of a small child's head, and it was just an incredibly stressful and very, very fast, steep and brutal learning curve. And the one person that perhaps kept me on the straight and narrow was Jane Shipsley, who was a wonderful clinician that I worked with, who really introduced me to the idea of doing this personalised medicine, of taking a very tailored approach in all the patients that we see. And whilst I learned absolutely the craft from the 2 masters at the top, the real nuances of medicine I started to learn from Jane. And what she encouraged me to do is to think about a residency, and I was very lucky to be able to go and do that in Cambridge. And I really remember the first time I went to Cambridge that I didn't really think people lived like this. I'd hardly lived from the ghetto in Hartlepool, but nonetheless, the northeast was very much my anchor point. And to go down and just see the glory, the splendour, the swagger, and just really the arrogance of Cambridge, it's really, really struck me, and it just seemed like an incredibly exciting and wonderful place to be. And so out of a cast really genuinely of hundreds, these would be the people that it's kind of nice to have an opportunity to say thank you to. And these are the people, Mike Heritage, Nick Jeffries, Ian McConnell and Steve O'Reilly. It all taught me many, many different things, but Mike was absolutely about the clinical finesse of phenotyping cases very skillfully. Nick Jeffries about trying to learn from every case that we see. Ian McConnell just from the sheer raw hunger and demand for excellence, both at the individual and group level. And then Steve O'Reilly, who perhaps had the most influence on me of all of these characters, that taught me an awful lot through this dog, Porridge. And a bit of a long story, but Porridge came in with a suspected history of hypergenocorticism, and it actually turned out that rather than having too much steroids, glucocorticoids, what it actually turned out is there was a complete deficiency. And this is something that we don't really view as being compatible with life, and yet here was this dog that would characterise for many months to have no discernible levels of glucocorticoids. And so really we were very intrigued with this, and I just wrote an open letter to Steve and the endocrine department at Addenbrooke's to ask for some help, and we got into an exchange, and he really absolutely opened up my eyes in terms of what the world can look like from high-end translational medicine. He was the man that really made the incredible breakthroughs about understanding obesity and the genetic cause of it, and moving it away from a disease of really just gluttony and absence of self-control to a disease that really was truly genetically driven. And through his work, he actually was able to identify an awful lot of the physiological controls of appetite. And his work with me in Porridge, where basically we suddenly started sequencing, we called in favours from absolutely all over the world to try and work out this, really showed me what medicine would look like from a human perspective. And from this time point on, I knew that I wanted to be like Steve. He was going to be the person that I was going to try and strive and emulate, and even if I never would meet his dizzying standards that he achieved, given the greatness that he has gone on and achieved, I wanted to at least try and begin that process. So our work really begins all the way back with Digby. So Digby was a dog that came in through the clinics that had very high levels of calcium, and so we were concerned with this, about it being a problem with his vitamin D status and his axis. And it really wasn't through the sort of family connections that I got into this whole area of research. It was very much from Digby as our first case that we saw. And I'm going to keep the talk very, very jargon-free for the last few slides, where really the process of vitamin D is taken in through the skin and also diet. And what we see here in this first pass metabolism in the liver, this is the 25-hydroxyvitamin D, which is sort of stable. This is the circulating metabolite of vitamin D. If you go and ask for vitamin D status to be assessed in yourself at your GP, that's what they'll go on to measure. And then this is the really active vitamin D metabolite just down at the bottom here. So what we were able to find in Digby's case is that he had this very high level of calcium, something that we'd be very, very concerned about. His PTH and PTHRP, so this is the parathyroid hormone, parathyroid-related protein, these were both very, very low. These are the only two other things that can cause a high calcium, so we were very, very concerned that he might have too much vitamin D. And to kind of cut a long story short, what we found out was that there's too much vitamin D being added to his food, and that's absolutely what he had wrong. So he just had too much vitamin D added to his diet, and that was what was causing the hypercalcemic state. And with the success of this case, it allowed us to start to look at calcium metabolism in a lot of other cases. And this is a dog called Poppy that we examined that had very enlarged lymph nodes and had what we call a granulomatous type of inflammation within the lymph nodes. And in this case, what we find here is that the calcium in this case was very, very high. And we showed in this case, it's by the active vitamin D metabolite being produced by the inflammation tissue shown in the slide that's then driving this hypercalcemic state. Another case that was extremely important to us was a dog called Hooch. And Hooch presented to us with a very severe form of IBD where he was losing an awful lot of protein from his intestines. So very similar to Crohn's or ulcerative colitis. It's a slightly different pathology, but the same sort of end product, that there's an awful lot of proteins and nutrients being lost across his intestines. And he was the first dog that I would really show that the vitamin D metabolism was absolutely disrupted in this case. And this dog was important because it led on to the first cohort or population study that we then went on to have a look at. And this is the very first study that I did when I came to Edinburgh. And really, I've got to say a big thank you to Adam for this, because for those of you who lived through this time, it sounds a bit trite to say this was a courageous study, but nonetheless, for those that lived through this time, that's probably a pretty appropriate word, because Adam risked an awful lot to try and help me get this study off the ground. And however simplistic and straightforward it actually is, it's very, very important for showing, well, this syndrome does happen in these dogs. We need to alter the way we treat them. And it allowed us to really start to think about vitamin D metabolism in this population a lot more. It then provided the basis for us to look at the relationship between vitamin D status and inflammation and mortality. And a huge number of clinical studies then flowed from this one paper that Adam very kindly helped with all those years ago. So one of the things that's very nice about this is we can now start to look at this across big populations. So this work within the clinics then allowed us to really expand our reach and start to think about examining vitamin D metabolism in a whole host of other populations. And a population that's always been very close to my heart is the St Kilda sheep study, which has been a great trigger point for a lot of these studies. And we now work with colleagues in Shrook to look at vitamin D metabolism in their Kirkton flock, which is having a lovely sort of validation type approach compared to our work in the very unmanaged St Kilda flock. We also do some projects that we all thoroughly love at the Edinburgh Zoo, particularly with the penguins, but also with the chimps and some of the other species that they have there at the moment. And then more recently, we've been able to move this into the African studies led by Mark Bronsford and Rebecca, the Ideal Herd, and we're now looking at the relationship between vitamin D and health outcomes in these populations as well. But I was just going to show a couple of slides on our St Kilda work, because this is where it all really started on the big population study. And I'm hugely indebted to both Ian Handel, Dan Nussey, Josephine Pemberton, and particularly Catherine Watt, who did a huge amount to get this study up and running. And this is a wonderful study for people interested in vitamin D biology because it's so unmanipulated. These sheep just live wild on this population, on this island population. They're completely unmanaged. It allows us to basically dissect out what vitamin D comes from the food, what comes from the skin. Because they're not given any additional food, we know it's entirely unmanipulated. They can't go to Malaga on holiday and get a bit of sun. They can't suddenly decide to want to eat lots of mackerel. They just have to get on with it on this tiny, tiny island. And it's been hugely informative for us as we've looked at the back catalogue of vitamin D statuses in these animals over this long period of time that they've been carefully stored down. We've now been able to tell an awful lot of interesting and stories that we think really informs the relationship between vitamin D status and health. And as an example of that, this was the very first study that was done by myself, Catherine, and the wider team that was really skillfully executed by Ian Handel, who's a wonderful statistician that we have within the team here. And this was the study that really made us interested about the relationship between vitamin D and health outcomes, because what we can see here on our vitamin D status and the number of lambs along here, it's almost a stepwise increase depending on the number of lambs born. So the ones that had the twins had a very much higher concentration than the ones that had the singles versus the ones that had no lambs at all. And this study, as we're now looking at it over numerous years, has been hugely informative to allow us to dissect out the relationship between vitamin D across a whole load of ecological parameters and situations. It's also allowed us to understand much more about the genetic and environmental controls of vitamin D. one of the things that's really striking about this is just the stability of the phenotype in this. So what we can see here on the sheep on the right here, they're almost the same. They're living on the same tiny island, yet they consistently have this very, very different levels of vitamin D. And from the genetic studies that we've done, we know this isn't really driven by the genes. There's something in the environment and the way these animals behave that really drives this very, very marked difference in their vitamin D status. And this thing about stability, it's again even in the penguins. The penguins have a pretty challenging time altering their UV exposure and their fish intake and this sort of thing when they're in the zoo. But nonetheless, we see the same stability of vitamin D status in almost whatever population that we've studied. What's really nice about working in vitamin D is it really is something that is easy to engage the wider media in. And so whenever we do stories on this, they quite often pop up and we get an awful lot of very respectable coverage. And I think this is one from the Times where they talk in very kind words about our work. But it's also picked up in our more esteemed journals as well. And this was the very exciting full-page spread that we had here. And as we head towards the ref impact cycle, I'll just flag this up to Charles. This is what the Sun says at the bottom here. So this is really only a couple of years away from governmental policy. That basically the government needs to pay for a couple of weeks' holiday for everyone in Scotland to make sure that we have enough taxpayers in the future. But probably more ominously, what we have here is the image of tombstones, which many people have pointed out. This is the way my career is going when I'm in hop with the Sun. Wherever I've walked, really, what I find really is the way's been cleared by Sir Edward. And where you think about when I'm talking about the merits of public nudity, there can't be many Mellanbys out of the pool of 120 that have been in the press discussing such matters. But what we find just up here on the top, Professor Mellanby was discussing this in the 1920s when he said it was no good to tell people in this country to go about naked in the sun to correct faulty diet. We agree. There's a prejudice against this procedure of this kind. Besides, there's so little sun. And again, what about the police? So, very sound advice then, very sound advice now. So just for the final few minutes, I was just going to talk a little bit about our work that really tries to extend from this story of vitamin D at the cross-sectional level that really talks about really this relationship in one point of time between vitamin D and then these range of health outcomes, to then thinking more mechanistically, well, this is just very nice, some nice associations, but what does it actually mean? And to do that, we've really got to start to examine this in, as we say, tractable and experimental systems to really define, well, what does vitamin D do? So why this is of interest to the wider field is that we've known for a long period of time is that your position latitudinally in the world can have a profound impact on your risk for development of certain diseases. And this is just a map of the US is obvious, but what we have at the top here in the more northern parts is this much higher incidence in these red colours here of MS than what we're seeing in the sunshine states of Florida and Texas. And this, along with a whole range of other lines of evidence, really suggests that vitamin D may be an important risk factor for the development of autoimmunity, with really MS being one of the lead things we're concerned about. And this is why, from our experimental point of view, we've really focused our efforts on understanding, from an MS point of view, what vitamin D does to experimental murine-based models of MS, which in this case we call experimental autoimmune encephalomyelitis. Now, we have lots of different ways of inducing this disease process. What we can take here is that we can seed the mice. These are our host mice here. We can seed them with cells that can only respond to myelin-basic protein. And this is a protein that's expressed in the central nervous system. And if we put this with a very strong adjuvant that really triggers and makes the immune system angry, what it can go on to do is cause this cause of disease that we can see here from the sort of flaccid tail through to hind limb paresis up here, this what we health where they develop clinical signs and then go on to respond, this monophasic cause of disease. We have another model system where we can take T cells and we can activate them in the lab and we can then put them back in. And these were all systems that were set up by Steve Anderton, and he very graciously and kindly during my fellowship years with him allowed me to interrogate these models with perspective from MS. And so in this system, we would activate the cells in the test tube with the peptide that they can respond to, with lots of pro-inflammatory cytokines, and we see a similar course of disease. Now, what we can see with this is that when we give high doses of vitamin D at these time points, we see a complete abrogation of disease with the active models. But when we take the passive disease models, what we can see in these scenarios is that given huge amounts of vitamin D, and in this case it's the active calcitriol vitamin D, we see none of this beneficial effect. It just doesn't seem to help. And so this has really anchored our group's long-term interest in trying to understand what happens between the two key protagonists of the activation of the immune system. And that really is the dendritic cell or the antigen-presenting cell, the myeloid cells, macrophages, call them what you want, but the cells that are able to interact with the T cells and then drive this pro-inflammatory response. And we've been particularly interested in understanding how vitamin D modulates the function and phenotype of this cell. So over the years, we've developed a lot of tools to try and interrogate this and to sort this out. This is just one of the examples that we've got, where in here we developed a very refined model of driving this experimental autoimmune encephalomyelitis, this model of MS. And in this, we take T cells that can respond to myelin-basic protein and put them in with a mice that has the dendritic cells loaded with the myelin-basic protein and then see whether they go on to cause disease. So it took a long time to work up this model, but after a lot of effort, we were able to do something where we can drive very respectable disease in this model. And it's very helpful for trying to work out, well, what does this cell, the master cell, have to do to this cell to drive pathogenic clinical signs? The other thing that's rather nice about this is, particularly from a veterinary angle, is we try and do wherever we can to minimize the impact and to improve the welfare of the animals in the systems that we do. And one of the nice side products of this was with the dendritic cell-driven disease, we don't see any inflammation where we inject the cells, whereas when we use the historical approach of using this really powerful adjuvant, we see this really quite nasty muscle necrosis. So as well as having a new model that really allows us to understand what these two cells have to do to each other to get disease, we can do this in a much more welfare-friendly way. And with Mark Vendrell, we've just recently published some work where we've validated, Mark has developed and we've subsequently validated the use of near-infrared probes, which again we can label T cells and look at them as they move around the body. And in here you can just see on the heats of animals that have had the cells versus ones that haven't. We can see these repeatedly over several days. We can see where these cells are going, where they're moving to as they come towards antigen and then dissipate around. And that's something that's been hugely helpful as we try and develop cell therapies to then try and see what happens to these cells when we put them back into animals. So as I mentioned, we're very interested to try and understand, well, what does vitamin D do to this one? So this is really the conductor. As we say it when we're doing these sorts of talks in Cambridge, this is a string quartet. In Glasgow, this is much more, this is the Ned, this is the policeman. So whatever your analogy is that you want, this is the boss man, and this is the one that's going on to do the damage. And so what we're very interested in is working out, well, what happens to this cell when it's exposed to high concentrations of vitamin D metabolites? And so again, helping Bruce out here with the big data, we can do big data type approaches here and have a look at gene expression. So this is the sort of the molecular machinery that controls the fate of cells. And what we can see is that when we take dendritic cells in their non-activated state that have been exposed to vitamin D, an awful lot of genes get downregulated and some smaller proportion get upregulated. And when we activate the dendritic cells, or the conductor, whatever you want to call it, we see again lots of genes go up and some, lots of genes go down and some go up. When we try to make sense of these data sets, what we try to do is basically try and make it ever more simple. So we've really focused our efforts on trying to understand, well, what happens between the cells that have both been activated and non-activated. And that cuts these huge numbers of genes down to just these 47 of interest. And we've been really interested to see, well, what happens to the ones that are increased in expression? Because we don't just want to study genes where basically the dendritic cells have been kept in this immature state by the vitamin D. We want to see, well, what is it proactively doing? And out of the 7 genes, the one we've really been interested in is this one, CD31, that we've spent a long time over the last couple of years trying to search. So driven by very skilled postdocs, and Iris and Louise, along with some really expert technical help from the QMRI and Pam Brown, we've developed a process by which we can manipulate the levels of CD31 in these cells. And what we can see here, where with this histogram plot, the further to the right, the more CD31 the cells are expressing. When we increase the expression of CD31, and these priming assays here, with pro-inflammatory cytokines along here, and just increasing concentration of cells here, what we can see is that when we increase the expression of CD31, these cells don't produce quite so much pro-inflammatory cytokines. And they don't have this capacity to drive this pro-inflammatory response. And so again, we've spent a long time trying to work out, well, why is this? And so just using some very straightforward microscopy techniques, we've been able to sort of show that when we take our dendritic cells here, we can see our dendritic cells and T cells coming into interactions, and we can then study them on these time phase microscopies to then look at the cells coming in, how much time do they spend. And what we've shown is that basically if they're increasing their expression of CD31, what we're seeing here is a failure of the cells to come together and spend time and basically be activated. So the increased expression of CD31 really pushes the cells away. And it's really sort of a very clever, interesting way upon which vitamin D can manipulate the phenotype of these cells to prevent the cells being able to activate the cells as they would do in the wild type situation. What's also rather nice about this is that this really takes us back at the full circle. So initially we were focused in on the work of the very first founder of the MRC, the first secretary, and really Sir John was the first person to really explore CD31 in leukocytes, and we borrowed on that work to show that vitamin D manipulates dendritic cell function, importantly through the CD31 axis. So, I think that's pretty much me. I just wanted to say about the future opportunities. Well, tremendous opportunities exist across the campus, and I take the responsibilities as an academic in here to try and make best use of those incredibly seriously. And one of the things that we've been able to do since the last few months is that we've been able to set up the vitamin D animal laboratory, which is part of this activity, offers a diagnostic service. And I have to pay tribute to Emma and Natalie, who've done a superb job of really getting this very complex machine that I would have taken an axe to a long time ago to give us really accurate metabolite measurements of vitamin D, which have now rolled out as almost as an international reference lab, and is something we're expanding ever more, the metabolites that we can measure. Teaming up with people like Jeff Schoenbeck, we can again bring this back into the patient context by really doing things such as whole genome sequencing, which we're planning to do on these two rather fantastic and very angry cats that have a syndrome of idiopathic hypercalcemia. And as two siblings, this gives us this really wonderful opportunity to really start and understand the genetic underpinnings of this disorder. And so really we're at a point for people that are interested in comparative medicine and translational medicine at this wonderfully exciting juncture where we're really making through the work of people like Dylan, who I'm sure will be in fullness of time very soon be doing this gig, who's a wonderful asset to this campus, who's really driving epidemiological studies in this space. Jeff Schoenbeck, who again is a wonderful asset to this campus, who's doing great things within the genetic underpinnings of disease and phenotype in animals. Taking that through into the diagnostics with the microRNA options that we now are starting to uncover, the flow cytometry-based work that we're now starting to do, as well as the proteomics and the whole genome sequencing facility. Again, tremendously exciting opportunities. And perhaps most importantly of all, we're now starting to see these being translated into treatments that are hopefully going to improve patient outcomes in the patients that we see in the clinics. And great, really imaginative, exciting things, particularly such as the gene editing options, are something we're very, very keen to try and progress in the months ahead. And underpinning all of this is the agenda of prevention that again has been very skillfully led by Stella Masri, who's one of the really fantastic assets that we have within the Roslin and a real star for the future, who's doing an amazing job of driving this programme of work forward. So I've tried to thank people along the way to avoid the massive list of Oscar-type thank yous, but I did just want to say a particular thank you to Professor Argyll, and I think now's the time to be gracious. I don't really want to talk too much about what everyone went through in 2011, but I don't think I was alone in finding that quite a difficult year, and it wasn't entirely sure whether my career was going to align more with the Argos storeroom, what was quite going to happen, and I've got Professor Argyll to very gratefully to thank for giving me a job. It's very, very much appreciated, and for all the opportunities that have come since. Also, a big thank you to Professor Anderton for his great support over a number of years down at the QMRI, and also to Richard O'Connor, who's wonderful to see back at the QMRI in the CIR, who's just a wonderful person and a wonderful scientist, and the CIR is very lucky to have him back. I'd also like to thank my own parents for all their support and help over all the years. As my father does point out from time to time, I've spent 19 years in higher education more than him, as I only had 18 months dabbling with the real world at Westways during that time period. And in the intervening years, him and my mother have done an amazing job of building up a hugely successful farm and business that's given myself and my brother tremendous opportunities. So for that, myself and my brother are very grateful. And also for the extended family and my Katie's trying to spit my words out. My mother-in-law and family here, and you know, tonight's absolutely dedicated to Richard, and he's a wonderful man, a man who just personified the life well lived. And finally, to my own family, on our recent trip to Genoa, trying to build careers in clinical science as a clinical specialist, as a translational scientist, and basically as a bench scientist. It's quite a time-consuming, fiddly job, even with just a modicum of success. And they're very patient as I live in this distracted fog most of my adult life. And I'm very, very grateful for all their patience and support over years. And I knew that bridge was dodgy when I drove over it. I could have told them that. So I have to give the last word to the man himself, Sir Edward. So I said right at the start, this issue of alcohol was something he was working on along at the same time. And so despite rickets being a really high-profile issue, working out ways upon which the armed forces wouldn't get hideously drunk was also a very important agenda. And this is something that Sir Edward was working on in parallel. And in what can only be described as the Journal of the North East, the spectacularly named British Journal of Inebriety, his work and his finest hours was reported in this. So he reports here that the most effective foodstuff in this respect is milk. Its specific influence in delay and absorption more than counterbalances its general effect as a fluid. And Dr. Mellanby comments upon the striking difference observed in the effects of a dose of alcohol when given 2 hours after the consumption of half a litre of water after half a litre of milk. In the first case, a dog may become incapable of standing or walking. In the latter, it shows no sign whatsoever of unsteadiness. So for those that can't resist the free bar, and I'm looking at you, Hickey, you know what to do now to avoid embarrassing yourself. Thank you very much.
Terrible flashback to Cleopatra in Glasgow.
Yeah.
Now, there was a mistake by the head of college at the start. It said that Richard graduated from Glasgow Medical School when it was Glasgow Veterinary School. A difference by at least 2 A-level grades. So what have we learned from Richard's lecture? Your time in Hartlepool makes my childhood growing up in the centre of Birmingham like the Riviera, so thank you very much for that. I think I can't understand why, after years travelling in the world, you've ended up in Thankerton, just south of Biggar. But I think more importantly, I think you've shown the importance of mentors and the importance of mentorship, and I know Richard does a huge amount of mentorship on this campus, and I'm hugely grateful for that. And I think that's because of the fantastic mentorship you have had while you've been both in practice and at your various universities. I thought what you eloquently showed there, Richard, was that don't underestimate the power of clinical observation, and we shouldn't forget that at all. And some of the work that you've done has been based upon clinical observation. You've gone from the patient to the population and back to the cell again, and that's absolutely fantastic, and that shows real importance. So I think with that, I just want to congratulate you on your inaugural lecture. I'm so glad I did give you that job. It was touch and go, but I'm really pleased that you stayed with us. So can everybody just congratulate Richard again for such